indicators. Substances which are neither acidic nor basic are chemically inert in nature. It is very harmful to touch or taste acids or bases. Hence, typical indicators are used to recognize them. The substances which change their colors in the presence of acid or base are called indicators. Now, let us study some indicators which are used in the laboratory. Laboratory indicators. Litmus paper is mainly used in the laboratory to test acid or base. It is made by an extract of lichen plant. It is blue or red in color. Blue litmus paper turns red in acid and remains blue in alkali, while red litmus paper remains the same in acid and turns blue in alkali. Similarly, methyl orange, phenolphthalein, and methyl red are used in the laboratory in solution form. Methyl orange indicator is orange in color and phenolphthalein is colorless. Methyl orange indicator becomes pink in acid and yellow in alkali, whereas phenolphthalein remains colorless in acid and becomes pink in alkali. Methyl red is red-colored indicator. It remains red in acid and becomes yellow in alkali. These are the various indicators which we use in the laboratory to indicate the acidic or basic substances. Universal indicator is one more type of acid-base indicator which is in the liquid state. It changes its color in the presence of an acid or a base. For example, originally palak extract is green in color, but after the addition of universal indicator, green color turns into blue color. Similarly, tomato is orange-red in color, but after addition of universal indicator, it becomes orange. If indicators are not available in the laboratory, then domestic indicators are used. Come, let us understand more about domestic indicators. Domestic indicators. On non-availability of laboratory indicators, natural indicators can be made by using several domestic substances. You must have seen yellow food stain turning red after washing with soap. This color change is due to chemical reaction between turmeric and alkaline material of soap. Here, turmeric acts as an indicator. Some other natural indicators can also be prepared from red cabbage, radish, tomato, and similarly from hibiscus and rose. Now let us perform an activity for making natural indicators. Making of natural indicators. To perform this activity, we require hibiscus, rose, turmeric, red cabbage leaves, white filter paper, etc. Let us begin with the activity. Activity. First, we shall take a hibiscus flower and rub its red petals on the white filter paper. Now, cut a strip of this filter paper. Red-colored paper strip so obtained is a hibiscus indicator paper. Similarly, let us rub rose petals on the white filter paper and cut a strip of the white filter paper. This red-colored paper strip is a rose indicator paper. Next, let us take turmeric powder in a dish and add small quantity of water in it. Now, cut a strip of the white filter paper and dip it in this turmeric water for a few minutes. After some time, remove the strip and allow it to dry. This yellow-colored paper strip obtained is turmeric indicator paper. Now let us take red cabbage leaves. Put these leaves in a china dish and add little water in it. Heat the content in the china dish with the help of a burner. Cool the solution and dip the white filter paper strip in it. Remove the strip and allow it to dry. In this way, we have prepared a red cabbage indicator paper. Now, with the help of these indicator papers, identify whether substances like lime juice, lime water, and vinegar are acidic or basic. Let us first take 3 turmeric papers and put few drops of lime juice, lime water, and vinegar on them respectively. What do you observe? We observe that the yellow color of turmeric paper turns red on addition of lime water, while it does not show any color change on addition of lime juice and vinegar. Next, let us take 3 hibiscus indicator papers and put drops of same substances on them respectively. What do you observe? Red color of hibiscus indicator paper turns magenta on addition of lime juice and vinegar, and green on addition of lime water. Similarly, take rose indicator papers and red cabbage indicator papers. Put few drops of lime juice, lime water and vinegar on them and observe the color change. Rose indicator papers show color change from red to pink on addition of lime juice and vinegar. It turns green on addition of lime water. Purple cabbage indicator paper shows deep red color on addition of lemon juice solution and vinegar, while it turns green on addition of lime water. From these observations, we can say that turmeric indicator paper shows red color in a base and remains the same in an acid. Hibiscus indicator paper shows magenta color in an acidic solution and green color in a basic solution. The rose indicator paper shows pink color in an acid and green color in a base. Red cabbage indicator shows deep red color in acidic solutions and green color in basic solutions. Hence, lime juice and vinegar are acidic, while lime water is basic in nature. Come, let us perform the next activity. Activity. Let us first take baking soda in a test tube and add small quantity of water in it. Shake the test tube and keep it aside. Now, take 4 test tubes and add samples of lime juice, vinegar, orange juice and apple juice in these test tubes. Next, let us add few drops of baking soda solution in these test tubes. What do you observe? We observe that effervescence are formed in each of these test tubes. These effervescences indicate that lime juice, vinegar, orange juice and apple juice contain acids in them. From this activity, we can say that effervescences are formed on addition of baking soda solution in the acidic solutions. By these simple activities, we can easily identify acidic or basic substances. Now let us perform one more activity to identify acidic or basic substances using laboratory indicators. Activity. We shall test solutions of vinegar, lime juice, ammonium hydroxide, dilute hydrochloric acid, and dilute nitric acid by using indicators like red litmus paper, blue litmus paper, phenolphthalein, and methyl orange. Observe the color change and note the observations in the observation table. Let us first test each of these solutions with red litmus paper. What do you observe? In vinegar, lime juice, dilute hydrochloric acid, and dilute nitric acid, red litmus paper does not show any color change. But in ammonium hydroxide solution, it turns blue. Now, let us test each of these solutions with the help of blue litmus paper. Can you tell which of the solutions will show color change? Vinegar, lime juice, dilute hydrochloric acid, and dilute nitric acid change the color of blue litmus paper to red. But ammonium hydroxide solution does not show any color change on the blue litmus paper. Further, take out 5 ml of each solution in different test tubes. Now let us add few drops of phenolphthalein in each test tube. And observe the color change. Phenolphthalein indicator does not show any color change in vinegar, lime juice, dilute hydrochloric acid, and dilute nitric acid, but it turns light pink in ammonium hydroxide. Similarly, take about 5 ml of each solution in different test tubes and add a few drops of methyl orange in each of them. What do you observe? In vinegar, lime juice, dilute hydrochloric acid, and dilute nitric acid, the red color of methyl orange changes to pink, while in ammonium hydroxide, it turns yellow. Do you know which solutions are acidic and which solutions are basic in nature? We know that. Blue litmus turns red in an acidic solution and red litmus turns blue in a basic solution. From this, we can conclude that vinegar, lime juice, dilute hydrochloric acid, and dilute nitric acid are acidic in nature, while ammonium hydroxide is basic in nature. Hence, we can say that phenolphthalein does not show any color change in an acidic solution, while it changes from colorless to pink in a basic solution. Methyl orange shows color change from orange to pink in an acidic solution and from orange to yellow in a basic solution. Now let us collect 3 soil samples from our surroundings in 3 different beakers labeled as A, B, and C and find out whether they are acidic, basic, or neutral. Let us first add some water in each of these beakers and mix properly. Filter each sample through the filter paper and collect the filtrate. Now test the filtrate in each of these beakers using blue and red litmus papers. In the first case, blue litmus paper turns red, while red litmus paper does not show any change. This shows that the first soil sample is acidic. In the second case, blue litmus paper does not show any change, while red litmus paper turns blue. This shows that the second soil sample is alkaline. In the third beaker, both litmus papers do not show any change, which shows that the third soil sample is neutral. Come, let us study about the properties and uses of acids.